Imagine a perfect match, a glove at a Berlin crime scene. DNA results, a 100% match. Case closed, or so it seems. But then the suspect's identical twin has the same genetic fingerprint. 2 men, 1 profile. Which one is the criminal? Welcome back, truth seekers. Across this series, we've followed DNA from the crime scene to ancient tombs, through shared lab battles with degraded DNA and the courtroom peaks of STR profiles. We explored ancient genomes and forensic genealogy. Today, in our finale, we confront DNA's dark side, the rare genetic twists that mislead justice and test the limits of molecular certainty. In 2009, a Berlin jewelry heist left behind a single glove. The DNA matched one twin from a pair of brothers, but also his identical twin. Both shared the same STR profile. The court had no way to tell who actually wore the glove. The case collapsed. DNA is our most trusted witness, but what if 2 people share the same voice? Identical twins are born from one fertilized egg that split in 2, meaning their nuclear DNA is almost identical. Modern science now tries to separate twins by sequencing their entire genomes and searching for somatic mutations, tiny changes that occur in cells. They might differ by just a handful of bases out of 3 billion. Somatic mutations arise through cell division errors after the zygote splits. Think of them as tiny post-it notes written after the original genetic blueprint was copied. Deep read sequencing and methylation patterns can sometimes pinpoint the culprit, but these methods are expensive and rarely used outside high-profile cases. Now, imagine the opposite problem. Not 2 people with 1 DNA, but 1 person with 2. Chimerism happens when an individual contains 2 distinct sets of DNA. Sometimes it's natural, embryos that fuse in the womb. Other times it's medical. In one real case from Alaska, a man who had received a bone marrow transplant for leukemia was tested for a crime. His blood DNA matched his donor, not him. Meanwhile, his sperm still carried his original DNA. To forensic scientists, it looked like 2 different men had committed the crime. Bone marrow produces blood cells. After a transplant, the recipient's circulating blood DNA reflects the donor's genome, but skin, hair, and semen still belong to the recipient's own genetic identity. These cases force labs to check medical records and use multiple tissue sources before declaring a match or exclusion. In a world of life-saving transplants and gene therapies, chimerism is no longer rare, and it's rewriting forensic protocols. Before DNA profiling, forensic serology used blood groups to link suspects, but this method came with a biochemical trap: secretor status. Roughly 80% of people are secretors, meaning their saliva and other bodily fluids contain the same ABO blood antigens as their blood. The rest, non-secretors, don't. Secretor status is controlled by the FUT2 gene. Non-secretors have mutations that silence the enzyme adding blood group sugars to body fluid proteins. For forensic scientists of that era, this meant false exclusions, a reminder that genetics can trick the very tests built upon it. In the 1980s USSR, investigators hunting the serial killer Andrei Chikatilo typed semen from crime scenes as blood group AB, while Chikatilo's own blood tested A. That mismatch helped him evade capture for years. Later DNA testing revealed the truth. He was the killer, and likely a non-secretor, his fluids lacking the expected antigens. Even today, genetic profiles aren't always unique. When a Y chromosome haplotype is rare or underrepresented in databases, a match might point to hundreds of distant relatives. Mitochondrial DNA and population substructure introduce statistical gray zones that require careful math to avoid overstating certainty. Forensics has learned to speak in probabilities, not absolutes. Every DNA profile comes with a match probability, sometimes 1 in a quadrillion, sometimes 1 in 1,000, reminding us that genetics reveals truth through statistics, not prophecy. From twins who share every repeat, to chimeras carrying 2 genomes, to secretors who silence their own clues, each story shows that DNA, our most trusted witness, can also be our most eloquent liar. Yet these very anomalies push science forward. They inspire better sequencing, deeper analysis, and a more ethical understanding of what identity really means. Our journey through forensic genetics ends here. but the story of DNA continues. Next, we'll turn our microscopes to paleogenetics and genealogy, unraveling how ancient and modern genomes connect us all. Until then, remember, in the world of genetics, even the truth has mutations. Make sure to subscribe to the Geneticist, like this video, and ring that bell to catch our next playlist on the genes that made history. See you soon.